So next up is Mora Regev, who did her undergraduate training in Israel and then did her PhD with Uri Hassan in Princeton. And I was very pleased to be able to get Mor to come here, actually funded via the Israeli CFAR program. So Mor is actually the Israeli fellow. I forget what your official title is, but it's something like that.
Some generous donor.
Yes, well, it's the Israeli.
Yeah, but there's another name, I think. Tannenbaum.
Tannenbaum,
Exactly.
thank you. That's a good thing we mention all the donors.
Yeah.
So, yes, so Mor will tell us about some of the work on intersubject correlation.
Yep. Thank you. Thanks for having me here today. Can you hear me well? Okay. Okay, so now that you met Sebastian and me, you might have noticed some perceptual similarities and also differences between us. We both have faces and two hands, but we differ in our height quite significantly and also the pitch of our voices. So if you would like to use brain activity to identify perceptual similarities or differences between us in your experience of the input, which is Sebastian and me, we might have a relatively easy time because by now we know pretty well where the sensory cortexes are and how they map. So maybe if we're interested in similarities in faces, we should check out the FFA. And if the pitch of our voices is of interest, maybe we should look at early auditory cortexes, right? But our experience is more than just the immediate sensory input, obviously. Very similar mental or cognitive experiences could be evoked from very different sensory input. So despite the difference in our voices, you might notice very similar scientific ideas actually appear across our talks. On the other hand, very different mental and cognitive experiences could be evoked within the context of the same sensory input. So some of you might find my talk very interesting and choose to attend every word I say, while others rather disengage and think about dinner, which is fine. But the point is that similarities and differences in high-level experiences and processes are quite more difficult to track. So they're less likely to be divided into spatialized modules of the brain. Oh, the colors are different. They're more often spread across different cortical regions. They might even evolve over time, which add another layer of complexity. So how can we track how we can identify those similarities and differences in high-level mental or cognitive processes that extend beyond the immediate sensory input? So I'll present today one approach to answer these kind of questions, in which we compare the complex pattern of bold activity across brains of different individuals that are engaged with relatively naturalistic stimuli, such as movies, spoken or written stories. I'll present some examples for the results we can obtain using those methods, all from studies recently performed in Uri Hassan's lab in Princeton. So I'll start by explaining about the method a bit. I know you're all practically, you know it very well by now, but I thought that maybe some people that I don't have experience with would benefit for some kind of walking through the different details of it. So instead of the traditional task versus baseline comparison of the average bold response within a voxel, in intersubject correlation, we compare across subject, across brain, complex bold patterns, either in the temporal domain or the spatial domain. So let's think about an example for a group of people, of subjects that watch a 10 minutes long movie. Starting with the temporal domain, here we'll take the full time course from one region in the brain, from one subject brain, and we'll compare it with this is going to be the 10 minutes long time course, right? The full time course of the movie. And we'll compare it to the full time course in the same region in another subject brain by performing Pearson correlation. So, of course, we have a group of subjects. We can do it across all subjects and get an average value of this correlation for that specific region. And this value gives us a measurement of how reliable the signal is in that region across different people. And of course, we can do it for different regions of the brain to get a map of those correlations, a map of the reliability of the signal. So when we see a reliable response across different people, that means that the synchronization, there's a meaningful signal there rather than noise, because the alignment between them, the synchronization, must come from some kind of a shared experience that align the signal to each other across different brains. So here the shared experience is the stimuli, the movie they watched in our example. And you can threshold those maps of correlation by using, for example, bootstrap analysis, where you scramble the time course before you perform the correlations. And for the spatial component, if we think again about those people who watch a movie, now we keep the full time course. We don't keep the full time course as is, but we'll average it over all the time samples within a movie to get one average value per voxel for every voxel in the brain. So then this will result in a spatial pattern for watching a movie in one subject brain. And this spatial pattern could be compared to the spatial pattern in another subject brain that watched that movie, again by performing Pearson correlation. And this could be performed in a searchlight where that is centered on every voxel in the brain, again yielding this kind of map of correlation, map of reliability of the spatial alignment across those different subjects. So one way to get statistical significance is by scrambling the labels of different kind of stimuli. So if, for example, they watch other movies as well, we scramble the labels, we'll create a null distributions of the correlations of between patterns from non-matching stimuli. But exactly like the temporal domain, also here, when we see a reliable response across different brains of different people, that means that there's some kind of course shared experience across these two different people or group of people. This is the basic principle. So I'll show you some examples, hope it will clarify some ideas. But I'll start with examples for how we can identify shared high-level experience that is evoked from very different sensory inputs, starting with the temporal domain. So in this study, we are interested in asking which regions of the brain are common to both the spoken and the written language system, and not just whether they're active when we read something or listen to someone speak, but whether they actually process this signal in the same fashion, in a similar way, regardless of the modality. So in this study, each subject either heard or viewed a spoken or written version of the same 7 minutes long story while undergoing an fMRI scan. In the written condition, words appear individually, one by one, in the center of the screen, in a rhythm that accurately matched to the timing of the words that appeared in the spoken version. Okay. A post-scan questionnaire assessed every subject's comprehension and engagement with that story, and we found no significant difference across these groups. So here, by comparing the signal, this temporally extended signal across the two different groups, we can identify not just whether a region is active in those two tasks, but whether the region processes those two different inputs in a similar way. So when we see this cross-group correlation, that means that it reveals synchronization that is driven specifically by the shared content, which is here, for example, the more abstract content of the story, rather than modality-specific kind of processing. Okay? So here are the results of this analysis. For example, we can see both linguistic and other high-level regions that show really demonstrate remarkable invariant to the modality of the story, showing the same kind of similar responses to the story, whether it was presented orally or visually originally. And just for illustration, we can focus on one region of interest, the precuneus, for example. You can see here that temporally extended signal on average, the average of two groups. In green, is it green here? It's supposed to be green, the readers, and red, the listeners. And you can see how it really follows each other nicely over the entire duration. This is not the case if we'll focus at the auditory cortex, for example, where the signal is not synchronized there. Let's move on and see another example for how we can see shared high-level experience that is evoked by very different kind of inputs, now in the spatial domain. This is a study led by Janice Chan, now in Johns Hopkins. And in this study, each subject watched a 50 minutes long movie in the scanner. It was the first episode of the BBC's Sherlock. So we have a Sherlock in common here. I just noticed now. And after that, they were asked to recall and describe in as many details as possible that movie with no visual cue or any experimental guidance how to do it. Okay? So each participant provided their own description of the movie that lasted from 10 to 40 minutes. So one of the questions in those studies was whether how similar the neural representation across different people when they recall that movie, despite the differences in the way they actually recall it. So here, the full bold signal from each subject's session of recall was divided into the different scenes of the movie, and then averaged over time within each of these voxels to create this spatial pattern. And then the correlations were performed across different subjects, across the patterns of matching scenes in the third slide, where the baseline was achieved by performing this correlation between patterns of non-matching scenes. So these analyses reveal this map that show regions. The regions show really scene-specific neural pattern that is shared across different people, regardless of the fact they didn't experience any stimuli when they recall and spoke out, and the really significant differences in their behavior when they recall the story. So we have here high-level regions throughout the default mode network. You can see, for example, the posterior medial cortexes. We also have high-level category-specific visual regions, but not low-level visual areas. So when we see this direct comparison of event-specific patterns of similarity across different brains reveals this, you might say, spatial organization of the neural representation underlying recollection that is shared across different people. Okay, let's see other examples, the opposite side. Now we're thinking about examples for how we can identify different high-level experiences that are evoked from the same or similar sensory inputs. I'm going to show you an example in the temporal domain because I actually am not familiar with any example in the spatial domain. But note that there's no reason why this kind of analysis could not be performed. I don't think it was done till now. So in this study, we ask which region of our brain might change the response to language, either spoken or written, as a function of top-down attention. In this experiment, in our two main experimental conditions, all the subjects were exposed to the same input. The input was two 15 minutes long stories presented simultaneously. One was spoken and one was written. The stories had nothing in common in terms of their content or rhythm of presentation. So the difference between our two main experimental groups is that one group was asked to attend the written story and ignore the spoken one, and I'll indicate the name of the group by showing the initials of the medium presented here, auditory and visual, underlying whatever is supposed to be attended, just for future slides. The other group did exactly the opposite. They were asked to attend the whoa, it's white. Oh, I guess you can see what I mean, right? The other group attended the spoken story, ignored the written one. In addition, we had two unimodal control groups of people who attended each story at a time. Group V just read a story with no distraction, and Group A listened to the story with nothing else to ignore. Wow, it looks so different. We instructed all the subjects to attend the story, pay careful attention to the details so they can answer a post-scan questionnaire that assessed their memory and comprehension of the story and get a bonus based on their performances. After the scan, we surprised them with additional questionnaire also about the story they were supposed to ignore. And we compared their behavior and saw that indeed they follow our instruction. They remember and comprehended very well the story they were supposed to, but they didn't remember anything from the other information. Okay, so now we have those two main groups of people who are exposed to two stories simultaneously, well, attending one of them. But how can we know which region of the brain is busy processing which of those two regions? So we can try to answer it by comparing this temporal signal with each of our unimodal control groups. So when we compare this signal to our unimodal group of listeners, because the shared experience across this group here is specifically the spoken story, right? This is the only thing in common here. The correlation that we'll get, the alignment, is specifically driven by that spoken content. And we can do the opposite by comparing our groups with our unimodal group of readers. And now what we're going to see, we can identify correlation that is specifically driven by the written information that is shared across the group. For the results of this analysis, starting with our group that attended spoken information, ignored written one. When we compare them to our unimodal group of listeners, we can identify those regions that process specifically that spoken content, and we see extended set of region from early auditory cortexes to high-level frontal and parietal region, as you might expect. But what happened next in the other scenario, where the same input was exactly the same, just attention was shifted away from that spoken content. In this case, we see that only auditory cortexes still continue to process that spoken story. What happened in this group? They were actually busy doing something else, right? They were attending the written information. So when we compare them now to our unimodal group of readers, we see that most regions of the brain were actually processing the written story from, again, early visual cortexes to high-level regions. But when this content was ignored, it was maintained within early visual cortexes. So this is an example for how we can use cross-subject correlation to identify regions that change the response as a function of the current cognitive demands. But we can try to extend this approach and try to track changes in how regions communicate, how they share information as a function of the current cognitive tasks. So a common method to trace shared information across brains is intersubject functional connectivity. Sorry, that's the punchline. Functional connectivity. Starting with functional connectivity, in which the time course from one region of the brain is correlated with other region of the brain, other region of the brain. So here, just for example, I'm showing you 61 independently defined regions of interest. For visualization purpose, they are placed here in a circle according to 6 independent networks in the brain. But you can do the same analysis on the voxel-wise. It doesn't really matter. So these correlations across different regions are performed in functional connectivity between regions within the brain of the same subject. So by this, the connectivity that we get, those correlations, are to a large extent intrinsic, and a lot of times they're relatively stable over time and across presentation of different kind of stimuli or different kind of cognitive state. But because we are here interested specifically to trace how information changes its routing or as a function of the current cognitive demand, we had to use another version of this analysis that we called intersubject functional correlation, ISFC, that was recently developed in Ulrich Hassan's lab. And in this analysis, we again compare the full time course from one region to other region of the brain, the same kind of correlation, but now we do it, instead of within the brain of the same person, we do it across brains of different participants. So now we get the pattern of connectivity we get is less intrinsic, but now more driven by the shared experience across the brains of different people. So again, we can do a very similar analysis to what we did before, just between different regions instead of within a region. When we compare our two groups that were exposed to 2 stories simultaneously with each of our groups, we can tag and know what kind of information is being shared. So if we compare our groups to our unimodal group of readers, that connectivity we'll get will be specific to our spoken written story that was attended, right? If we'll compare our groups to our unimodal group of listeners, we'll get connectivity that is driven specifically by sharing information from the spoken story. So here are the results of this analysis, again starting with our group who attended spoken story, ignored the written one. When you compare them to our unimodal group of listener, we see really almost most of the regions we have share information from that attended spoken story. But again, when this content is now ignored, although the input stays exactly the same, we see that it's mostly regions within the auditory network that still continue to share this kind of information. And again, we see the opposite effect. Region information is shared all over the cortexes, from low-level sensory region to high-level cognitive ones, but when it is ignored, it is contained within early visual regions. So overall, when you attend one story, ignoring the other, right, most regions of the brain, up to high-level regions, share the content of that attended story, but at the same time, low-level sensory cortex could still share information from the ignored content. So just to conclude, I showed you today how some examples for how we can detect brain activity that is specific to some kind of experience by comparing complex pattern of brain activity across people who share that experience. And when we have those complex patterns in one region of one subject brain and one group of subject could serve as a model for the response we can see in other subject brain. And that kind of saves us the need from creating an explicit model for mental and cognitive experiences, right? We can explore, it allows us to explore high-level regions that are usually a bit more challenging to model, and we don't need to have this really a priori assumption of how each region of the brain is supposed to participate in the processing of very complex experiences. It also allows you to use relatively naturalistic stimuli, such as narratives that extend over time and therefore are more likely to create a richer experience, richer specifically cognitive and mental experience. So I showed you only fMRI data today, I apologize. But intersubject correlation, as you saw yesterday, could be performed and was performed in other spatial and temporal scales, from EEG to ECOG, MEG, fNIRS, and just continue. These are just a few examples. So this could be performed in all of this. Of course, there are some advantages and disadvantages that we can discuss for every kind of method, and maybe some adjustments need to be for each of those tools, but the same idea, the same principle of that shared experience still valid for all those methods. That's it. I want to thank my previous and current advisors and people I've worked with, and for you for listening. Thank you.
Thank you, Mor. We have time for a few questions.
I really like your design, and that was very interesting.
Oh, thank you.
First a simple question and then a harder question. The simple question, just sure I understand. It's always a between subject design. One either listens or reads, but not both to the same material.
Exactly. You're talking about the last experiment I showed of the attention?
Any of them? Were any of them within subjects designs?
No, what I showed you, no. You could, no, it's all intersubject.
You'd have to do it with a different text, yeah, to avoid.
Well, you need to do it actually with this. You always have, when you're doing a cross-subject, you need to have, although you're using different stimuli theoretically, one is written, one is spoken, so on, you're still tracking some kind of shared component of the stimuli, right? So it could be just the story, that they hear exactly the same story, and this is what you're tracing in the correlation. But it could be some other thing, maybe it's a sensory component, right? It could be different. You can isolate shared experience, depends what you're looking for.
Okay,
right?
so I really like your technique for isolating the shared experiences.
Yeah.
I'm wondering about the underpinnings of reading, where subvocalization, auditory imagery may be playing a
Yeah.
role, versus listening.
Yeah.
And if you've tried surrogate analyses to contrast the correct listener with the other readers versus the correct reader with the other listeners.
What do you mean by the correct reader or
Surrogate analyses,
Yeah.
Repairing of the
Yeah.
inter-subject data.
repairing it? You mean cross groups?
Yes.
Oh yeah. So if we do cross groups in different, if you have two stories in different modalities and completely different content, if you compare them, you see no correlation. So I have this slide, but you'll see no reliable correlation because maybe I didn't mention it when I described the methods, but if you're doing, for example, cross-correlation across people who just rested with nothing shared between them, each person thinking about something else, you won't see any reliable correlations between them. So this is the same case. When I compared the people who read one story and listen to completely another, there are no significant correlation between them.
I'm asking for the case where they read or hear the same
The
story.
same thing. Oh, of course, this is the analysis I showed. I'm comparing readers to listeners, but there's only one story, so I don't have, right?
One could take each reader and contrast them with the other listeners. One could take each listener and contrast them with the other readers in a surrogate pairing reanalysis to test the relationship. You've pointed to the correlation between within listeners and within readers.
No, this is not within listeners, within readers. This is all across the two groups. So everything I'm showing here is between a reader and the average of the listeners. So the shared, okay, we didn't understand. So the shared experience here is only the general context of the story and not anything that is modality specific. But you can do it also, as you suggest, we can do it also across different subjects within the same subject. So, and I did it across different listeners and different readers, and you get two different maps, right? They're specific to the shared experience, but then you'll have the component of the modality in addition, right?
Two questions. First, why did your connection work?
Because it's Women's Day.
Okay, has anybody... I can live with this argument. I see this. Second question. Let's forget about the
You can
first.
only go down from here.
Yes. Has anybody from your lab or you ever thought about presenting the same stimulus visually and auditory at the same time, but let them either mainly focus on the visual or on the auditory domain and see how connectivity changes basically between visual and auditory? I mean, intersubject connectivity between visual and auditory cortices should flip or should switch when you have the same visual input or a different visual input. I mean, when you have one group with having the same story visually and auditory, the other group has the different stories visually and auditory, you would expect a nice...
So you're saying you're expecting when you have the same story in the two different modalities, you're talking about you'll see some kind of synchronization across the different modalities?
Yes, and a direction, I guess,
right.
Depending on where you focus your attention on.
If you're focusing, well, I expect a lot of synchronization anyway, in this case, between the visual and the auditory modality that are kind of, I don't know if we call it fake, but it's not necessarily direct connectivity, as we like to call it, the long-range axonal kind of anatomical, because they're locked, because the spoken story and the written story are presented exactly in a synchronized way. So just because of the stimuli, those two sensory cortexes will be correlated to each other, right? So I didn't show a control here, but I'm showing here that the reason I'm showing there's correlation in this and the other experiment between reading and listening of the same story is not because of the rhythm of presentation, but I actually scrambled words, the letters and so on to make sure that the similarity that we see is from the more abstract content or the linguistic content of the story and not the rhythm of presentation. So it could be, we can disentangle it. I must say that when I performed that experiment, it was like 2013, so we didn't have this approach yet. But I definitely considered going back and trying to play with this data using intersubject function correlation. Yeah.